Little shout out if it's in the weight room or did you set
It
it so they'll
should
just join?
it should just show me and I have to admit people, but I have it just mirroring display, so we should still be able to see it once the slides start, hopefully. Okay. Now I need to get actually back to presenting. No, do not show Zoom present view. Okay, this should now be right. All right, so good morning, everyone. I think everyone knows that I'm Justin Savage. I guess people on Zoom might not. I'm a 6-year PhD student in Chala Aragu's lab, and this is our Duke Quantitative Living Systems Center trainee breakfast. This is kind of a random topic that I've been wanting to present in some way for a while. It started out as being about genome browsers and then stopped being about that very much, but I'm going to talk about sort of just generally how you can work with DNA sequences and design plasmids using Python. And the specific example we'll use is for CRISPR-Cas9 cloning, but it can be kind of used more generally for anything that's kind of genomics oriented or like plasmid oriented. Um, but the major steps in the CRISPR sort of cloning version, which I'll go over sort of the way you can do this computationally, but this could also be helpful if you just want to understand how this process works. You design a guide RNA to target a gene of interest, which I'll explain in the next slide. You order oligonucleotides for cloning that guide RNA into a plasmid backbone. You verify your cloning with plasmid sequencing of some sort. You then test the guide RNA plasmid in a cell type of interest, and then design one of the easiest things to do is design a PCR strategy to amplify the guide RNA target region, and then you can validate the gene editing at that target region through sequencing. But just in case you're like very unfamiliar with these techniques, AdGene is a organization that's sort of a repository for different plasmids and viral vectors, and they make these series of ebooks that are like their 101 series. They're really great and have like a great introduction to each of these things. So they have one on plasmids, one on CRISPR itself, one on viral vectors. So if these are new to you, these are really great resources for that. And I'll borrow a little bit of their material from that, but definitely check them out. They've also recently they've made multiple versions and they're better than they were even a few years ago. So definitely recommend those. The specific example of a CRISPR-Cas9 system that I'll talk about is one developed by our lab called Gearbox. Here is the eLife reviewed preprint of that. But basically, Gearbox is a AAV vector that has a U6 promoter that expresses a guide RNA um, and a astrocyte-specific promoter called GFA ABC1D that expresses Cre recombinase with this fancy microRNA targeting cassette that makes it more astrocyte specific. But the basic idea is that every cell that gets the virus expresses the guide RNA, and only astrocytes express Cre recombinase, which activates expression of the required protein Cas9, which I'll also explain in a minute. But we have a lot of details on that um, vector and how to use it and how to do the cloning and everything that are linked in that article if you want to see like further details. But this is a specific case for gene editing in astrocytes, but pretty much every CRISPR-Cas9 tool uses a U6 promoter and a guide RNA of some sort. So that part is pretty universal. I'll also talk about Biopython, which is a sort of a collection of tools for working with sequences in Python. I didn't put a lot of links to those, but they have a website, biopython.org. One of the main classes is the seq class that they define well. And the other one of the tools I'll talk about is pairwise alignment. They have a good section on that. So if you need more details on those things, they're there. So going back to this, the first step for this CRISPR cloning is to design a guide RNA to the target gene of interest. So the way that CRISPR-Cas9 works is that the Cas9 enzyme needs to be present, and then you have this thing called a guide RNA. The guide RNA has sort of two parts. There's this scaffold, and that's going to be the same for every guide RNA. It just helps the RNA to bind to the Cas9 enzyme itself. And then you have this spacer region, and that's the That's the sequence that's actually specific to your gene of interest. So that's the part that's going to change. And when these come together, they make this Cas9 guide RNA complex, and that's what's able to actually bind to DNA and edit it. The way this works is that there's this PAM sequence, which means protospacer adjacent motif, which basically just means it's beside the spacer. This sequence isn't actually in your guide RNA, but the Cas9 enzyme only checks at these PAM sequences for matches to the guide RNA. So this is required to be in the genomic sequence, but not in the actual guide sequence. Once Cas9 recognizes that and this complex forms with the DNA, it will cause a double-stranded break in the normal version of Cas9. There's also a lot of other versions do lots of other really cool things. And then the DNA will repair through one of two mechanisms, either non-homologous end joining, which is a sort of more error-prone version, or homology-directed repair, where there's some sort of donor DNA that can be repaired with. If you've added some sort of DNA for some, like an insertion, the cell can use DNA that's around to make a repair, and that's a way that you can insert a sequence of interest. If you don't have anything like that, what will happen is that you'll get a double-strand break, and most of the time, the cell will actually be able to repair it correctly. But if there's a correct repair, Cas9 is still there, the guide's still there, it's going to cut again. So you're having this repeated cut and repair until eventually the cell makes a mistake. And if the cell makes a mistake in a protein coding gene, it will cause usually a frameshift mutation that will then make the protein product incorrect. So like I mentioned, you have this protospacer region. That's what we usually like talk about when we're talking about the guide RNA. It's usually 20 base pairs, and that's a sequence that matches the genomic target like exactly. And then you have this protospacer adjacent motif, which is in the genomic target, but not going to be included in the actual plasmid, but it is required for the targeting to work properly. The exact sequence of the PAM depends on the species of Cas9 that you're working with. So the most common one, and if like people don't say anything about it, they're probably talking about SP-Cas9, which is the more common variant people have used. Its PAM sequence is NGG. So it's basically any nucleotide and then two Gs. So that occurs a lot in the genome. It's a pretty short sequence. The other version that people use a lot is SA-Cas9, and these letters are just the bacterial species that the Cas9 originates from. It's a smaller variant that can be more easily packaged. So you'll usually see this for approaches where they're trying to put the Cas9 and the guide RNA in the same plasmid. That works well. Its PAM sequence is a little longer and more complicated, with NNGRRT, where R is an A or a G. So it's less likely that you'll find a good SA-Cas9 site wherever you're looking to knock out. But there have been really good uses of either approach. Something I really want to emphasize is if you find a guide RNA for your gene of interest in the literature, make sure you know what species it was, because most of the time they're not going to be interchangeable. Um, some of them could be because you do have the N and G. So if you just get lucky, you could find one that works for both. But make sure that you check carefully to make sure the PAM is correct for the version of Cas9 that you're actually using. There's also a lot of other flavors of Cas9 that I won't get into that people have made. But these are by far the two most common. So for picking this actual target region and designing your guide, the first thing I would say that's not like quantitative at all is to check the literature for guide RNAs against your target. If someone has already made a really good guide RNA against your gene of interest, it's going to be way better to use the work they already did than to start from scratch. The other thing is that it's kind of hard for these algorithms to take into account is a deep biological understanding of your gene of interest really helps you for all of these techniques. If your gene has a lot of transcriptional isoforms, a lot of these programs aren't going to understand that. Or if your gene has a really high protein stability, even if you knock it out, it might not make a functional difference in the cell. So you need to keep that sort of biological understanding on your side as well. Another good thing to do is to check the target gene on a genome browser, and that's a good way to tell if it has a lot of transcriptional isoforms. So my favorite is this one from UCSC. They have a genome browser for pretty much every species, and they have I won't go into them much here, but this tools tab on their website also has a lot of really useful tools that can do a lot of the things we're going to talk about in a web interface, if that's easier for you. But if you look up a gene, for example, this is beta-actin in the mouse MM39 genome. Um, these different annotations here are showing different transcriptional isoforms that have been identified for this gene. And these little boxes are the exons, and the regions between them are the introns. So one thing you want to focus on when picking a region to target is that it's actually expressed in all of the isoforms that you care about. So for example, if we pick this second exon, you can see that there are some of these isoforms that don't include it. So you could have protein being produced from those transcriptional isoforms, even if you have a successful knockout in that region. So just something important to keep in mind. The other thing, of course, is if you're introducing a frame shift, you want to be early on in the coding sequence. If you have it at the end, you might still be producing functional protein from that earlier part of the sequence. And similarly, you don't necessarily want to just pick the first exon every time because that's often there will be an alternative start site for transcription that could then take over if you target the first exon. So um, the more you know about your gene, the more intelligently you can design these, but generally towards the beginning and focus on exons that are important. You don't necessarily have to knock out every exon. You could definitely make experiments where you only knock out particular isoforms. Those could be really interesting, but having more of a biological understanding of your gene is really helpful. So there are two really good websites for this. One is CRISPR, which is from the Broad Institute, and we've used that one a lot. The other that I really like is Benchling, which is a online lab notebook slash plasmid editing tool that's free for academics. I really like the way they do it because if you upload a DNA sequence, they'll show you guide RNAs along the sequence with different scores, so it's easy to see like exactly where they're going to be. A lot of the other tools don't really do it that way. Um, and then the other one I like is called GuideScan, and that's what we'll focus on today. The reason that this one's useful is that it has a command line version for doing sort of in a higher throughput way and like algorithmically. Um, there's a new version of GuideScan that didn't have time to incorporate into the presentation, but in the original version, Charlie Gerschbach's lab from the biomedical engineering department here has a good protocol for that I've linked here. And I'll just briefly talk about how they set that up. Um, for pretty much all of these, the first step is to create a condo environment. Some of the ones I'll show later are in Google Colab, so then you wouldn't have to do that. But I did a session, I guess maybe 6 weeks ago on that, so that's linked here if you need a refresher on that. Um, there's also detailed instructions in that paper from the Gershbach lab. But basically, the way that this works once you have GuideScan installed is that you run this command called GuideScan guide query. And there's a few different things you input. So this -b is a file that is for your both your Cas9 and for the species of interest. So they have several available on their website. GuideScan does. So this one I had the Cas9 MM10 file. They have several for different combinations. If you're working with like a less used model organism, they also have instructions for making your own. It's just a little bit more involved. The other thing that you'll use is a file to tell it where you actually want to make guide RNAs against. For this case, you can give a bed file, which is a file which lists genomic loci. I think we actually also talked about bed files in the epigenomics analysis one that we had like a month ago. But a bed file is basically just a list of genomic coordinates. So each genomic coordinate that you give, it will design a guide RNA against. So one advantage of this technique is you can list, as many as you want. The Gershbach lab recommends using this command line version if you have over 1,000 targets. So you can, imagine like a lot of these. The other thing that's nice for this is these don't have to be gene loci. These could be like an exon you care about, or the Gershbach lab does a lot of work with enzymatically dead versions of Cas9 where you can edit the epigenome instead of the genome. And for those, then you could enter, regions you think might be enhancers or promoters or, any sort of genomic element. It doesn't have to be a gene. So this gives you a lot of power to make these sort of guide RNAs. And this would be most appropriate probably for like a higher throughput like screen where you're going to be doing like single cell sequencing or things like that. So I won't get into those techniques much, but definitely can check. They have some information about that in the Gershbach Labs protocol, as well as just their work is really useful example of all that. All right, so what you'll get out of this is these 20 base pair guide RNA sequences. So once you have that, you need to actually order DNA to make that into your plasmid backbone. So the way that is most commonly used to clone these things in is with a sticky end cloning, where you would digest the backbone, which is that plasmid that we originally started with that has the U6 promoter and usually the guide RNA scaffold on one side. So you'll break that apart at that point. And usually this is done with a restriction enzyme because that just makes it a lot easier. So these backbones are usually designed with, this cloning in mind. You'll then insert your guide RNA with complementary sticky ends, which I'll illustrate in a second, so that it like perfectly fits into that gap that you created, and then usually ligate it using something like T4 ligase. So this is the exact sequence for how this works in Gearbox. Um, Off to the left of the screen would be the U6 promoter, and off to the right of the screen would be the guide RNA scaffold that helps it to bind to the Cas9 enzyme. So Gearbox has two SAP1 recognition sites that are in this region. So when you digest with SAP1, it cuts outside of its recognition sequence here and here. The reason that Dinesh, a former postdoc in our lab, designed it this way is that once this cuts, it will actually remove this region here. So you've physically removed the SAP1 binding sites. And these sticky ends are not complementary to each other. So it makes it so that religation of the plasmid to itself is unlikely. So that's pretty common for a lot of these techniques and that it increases your cloning efficiency. But in any case, we've removed this piece of the DNA that was between these two sites, and we're left with, because the enzyme cuts sort of at an angle, we're left with these sticky ends where most of the DNA is double-stranded, but you have this extra piece that's only on one strand or the other. So then we'll design guide RNAs that have complementary sticky ends. Um, and you'll have this kind of conserved sequence that matches the sequence in the backbone, and then you'll have the guide RNA sequence that is unique for each of your guide RNAs against this given target, and then the reverse complement of the guide RNA so that it's double-stranded. And then those will sort of just anneal in there with the T4 ligase. You can sort of see these two bolded ones. In the gearbox strategy, we have an extra nucleotide on each end in the sticky end. I don't have a good biological explanation for that, but Dinesh tried it and it made this cloning a lot more efficient. So I don't know if this extra nucleotide gets sheared off or something, but having this extra nucleotide increased the efficiency for this particular cloning. But in general, you probably wouldn't include that. So the way that we can actually get this DNA in hand is to order an oligonucleotide that has this forward sequence, and then to order another one that has this sort of opposite strand sequence. So the actual like algorithmic steps for making that would be to take the guide RNA that we got from the either the website or from GuideScan, to add those sticky ends to the forward guide RNA sequence to match whatever backbone you're working with. You then would generate the reverse complement of the guide RNA and add the sticky ends that are appropriate for that version. And then you want to output some sort of format that can be used for the actual ordering. So a lot of people in our lab do all of this with like Excel and stuff. I know that I would make a mistake with all that copy and pasting and like going from one website to another and all of these things. So the way that I do this is with Google Colab and with Biopython. So in Google Colab, which I have the link here if anyone wants to use this or play around with it later, it'll be available on the website. But I call this one guide overhanger. So the first thing is for Google Colab, to import a file, you can use this Colab files like package. And then here I just upload a Excel sheet that has a name for each guide RNA and the actual 20 base pair sequence. You can then install Biopython on your like instance of Google Colab using pip install Biopython. If you include this exclamation point at the beginning, it's running basically a Linux command line command instead of actual Python code. And then we want to import from Biopython this seq package, and then pandas, which is used a lot in Python for working with sort of spreadsheet or tabular data, and then the files thing, which I guess I'm importing twice for no reason. Um, then I read in this Excel sheet using pandas, and you just have to have these two columns that are the guide name and the guide sequence, which I then make a list in Python. I then have this function that I wrote for doing that computation we talked about. So if you have your forward oligo and we know we want the guide RNA sequence plus that CACCG, if these are in the form of Python strings, we can just add them together and it will do a string concatenation, which is what we want. Um, I then create this seq variable, which is an actual like BioPython seq object of the guide RNA. So this is going to recruit or create this sequencing object using the guide RNA string that I put in. So then once it's a seq object, it has these extra methods attached to it where Python understands this is now like a DNA sequence. So then when we want to make this reverse oligo, we can take the AAAC that we need to add on and then add this seq dot reverse complement. So we'll intelligently know this is a DNA sequence and I need to produce the reverse complement. I then use str to convert that back to a string so that the concatenation will work normally. And then we can add that other C onto the end. And then because it's all happening in the function, I'll return the forward oligo that we made here and the reverse oligo that we made there as a string. I guess I haven't stopped for questions at all. Are there any questions so far? Cool. So then I just make an empty list for saving all of the forward oligos and the reverse oligos, and then have a for loop for the length of the list of guide RNAs that I put into this. And then I use that function to make the oligos for the forward and reverse of each guide, and then append that to that list I created at the beginning. And then for saving this outward, I make it a dictionary, which basically just kind of prepares it for these lists to be like have their own names. So guide name is the name for basically the column that is in guide names, and guide sequence for guide lists and forwards and reverses. And then if you have a dictionary, pandas can convert that to a data frame, which is basically a spreadsheet. And then with pandas, when you have a data frame, you can use this dot to Excel, and that'll actually like save an Excel file that you can put out. And then because we have this Google Colabs files package, files.download will like actually download this file to your local computer when you run that line. So then we have an actual Excel sheet that has our guides and then the oligos we would actually order. The next thing that I run is kind of a cleaning up of this same file. So I put all of the forward and reverse sequences together into one list. And then I append to their names either forward or reverse, basically. And then I make a new dictionary that has the name, sequence, scale, and purification columns. And the reason is because IDT, that synthesizes oligonucleotides, will take a file of that format. So if you have name and then have any appropriate string, sequence, and then the sequence of nucleotides, and then the typical scale that we use is 25 nanomoles, and then standard purification. If you make that into an Excel sheet, that you can upload directly to the IDT website and order those. So even if you were making 200 oligonucleotides, they'll send them to you in 96 well plates. So you could order, a ton of these at once with a sheet like this instead of like doing this copy pasting thing. So this is, a helpful way that I do that, and you can order these more quickly. I save it in both ways because I like in the original file format that you have the forward and reverse sequences together, and you get a little bit more information. So I would save that kind of thing to my lab notebook so I know like what the oligos were. But then this format is a lot easier for that ordering process. So now that we've ordered oligonucleotides, and I'm just going to assume that we cloned them in successfully, that procedure actually works really well most of the time. You would then want to verify that that cloning worked with plasmid sequencing. Um, there's sort of two easy ways to do this. We do this with GeneWiz, which is part of the Azenta company, but there's a lot of different versions of that. You can do either U6 primer Sanger sequencing or whole plasmid sequencing, which there are several companies doing now with Oxford nanopore technology. But one of the things that's nice about pretty much all CRISPR-Cas9 systems is that they'll use a U6 promoter for the guide RNA. And most of these companies will have what GeneWiz calls like a universal primer, where they already have a primer for Sanger sequencing for U6. So if you send them DNA, they will do that sequencing for you without you needing to send them a primer or design a primer or anything like that. So it's almost universal that you can sequence that your guide RNA was cloned into the proper location with that Sanger sequencing. And then I just validate that with the Python version. So here things are pretty similar for the setup. The thing that I change here is I import from Biopython this seq.io part of the package. This is for actually like reading in files that are in a sort of like a sequencing format. I also import OS, which helps you kind of work with files generally in Python. Um, I then read in the Excel file. I did this in a bit more complex syntax because if you've uploaded a lot of different files, there might be some that don't have Excel endings. Earlier, we were only uploading one file, so a little easier. So this is basically just looking in um, the current directory of Google Colab for files that end in XLSX. I'm then going to read in the first file that ends in XLSX. Hopefully, we've only imported one, and that's that file that has the guide RNA sequences, and then we turn it to a list like we did before. I then want to make a reverse complement for each. I'm going to search for these guide RNAs in the sequencing file, but if your sequencing was in the reverse complement orientation, you won't actually be able to tell that. The function I'll use just literally searches, so it's not going to understand reverse complement. So I just make a list with both, and I search for either the forward sequence or the reverse complement. So I just add that as if it was like an extra guide, just with the name with RC appended to it. So then I upload these sequencing files from GeneWiz. You can download this .seq file. I don't know why they call it .seq because it's in FASTA format, which is like a standardized genomics format. But they call it .seq, which is fine. It still works. I then have this function that I wrote that's a little superfluous for doing the reading, but the gist of it is it's using this seq.io parse, which is going to read in the sequencing file. The basic idea is that when you tell it it's a FASTA format, the first line of a FASTA is like metadata basically, so it's going to ignore that and then read the actual sequence as one line, even if it ends up appearing in multiple lines. So then I get the file list for all of the files that end with this .seq, and then I make an empty list to store them, and then have this for loop where I'm reading in each of these and calling them this FASTA format, and appending them to this list that I created. So then you have this list of the actual sequencing results you had. For Sanger sequencing, this will usually be like 600 to 1,000 base pairs of sequence, depending on how good it was. For Gearbox, the guide RNA is only about 60 base pairs in, so you will definitely get a good read through that. I then have this sort of more complicated for loop. I'm going to focus in on just the key part of it here, where for, again, the length of the guide list, I'm gonna say the current guide is guide list I, just give you that first one, or whichever one we are based on using I to iterate. The current name is that same order in the name list. And then I'm gonna take seek result. The zeroth item in this seek result like object is which is just that file that we read in, is the actual sequence, because it reads it in as a seek object. So we just want the sequence as like a string. I'll then use .seek.find and the current guide. So this is just going to like literally search for that exact match sequence in the other sequence. So it's not doing like a real alignment. It's not going to accept any sort of mismatches. It's just going to look for the literal sequence to be there. And then it's going to tell you the index along the sequence where the guide RNA showed up. So I just say if it's greater than 0, I want to actually add that to something to print. If it's 0, it means it never found it. Or I think it actually returns -1 if it never found it, because 0 would be the first item. I think it's -1. Yeah, greater than or equal to 0 means it's -1. So then I print that out. So just to show a little bit of the housekeeping before, I make an empty list for each of these things. I'm doing this for each of these sequencing results, which was just the like .seq object version of the files that I read um, in that seq objects list. Um, I'm gonna append the name and the sequence. If you do seq result 0-ID, you get the name of that file. And then if you do .seq, you get the literal sequence, what we learned used later. I then turn that into a data frame again so that it can be an Excel sheet. And then we print it out. I meant to have a slide of the actual output of this. But what this will print is the name of the sequence that's here. It'll then print the name of the guide RNA that you had and the actual guide RNA sequence. So you'll get a print of which of your sequence files actually had the guide RNA or one of the guide RNAs that you cared about and which one it was. So even if you like accidentally cloned the wrong guide RNA into the other one, you can kind of see that there. The other alternative is that you did whole plasmid sequencing. So for this, you're going to get an actual plasmid map for the entire plasmid. This is nice to have to know that when you were doing this cloning, you didn't accidentally mess up some other part of the plasmid, such as like that Cre recombinase or the actual U6 promoter itself. So here you can upload the reference map, which would be like the map that is you would kind of expect to have, and then a new map that would be the map that you got from the sequencing. GeneWiz will return these as a GenBank format, so you can say the format is GB, but that's like a normal plasmid map that you might have worked with Benchling or Ape or SnapGene or those kind of tools. And once you read both of those in, we'll then use this Biopython aligner package to do an alignment. So this makes an object that I'm calling aligner, and the aligner like variable basically just stores the parameters that you want to use for the alignment. Here I'm doing a pairwise alignment, meaning that we're only going to do two things. We're not doing like sequence alignment or multiple sequence alignment, like if you did, RNA-seq or something. The match score is just saying that every time you have a match, give yourself 1 point. It's basically you always say that it's 1. The aligner mode, by default, it's going to do a global alignment, which isn't what you necessarily want for this. So global alignment is trying to find like anywhere in the sequence it can make good matches. So what it ends up doing is if you have like a shorter sequence, especially if it's like Sanger or something, it's just going to like spread it out to match, which is not exact what you want at all. So local alignment is saying like reasonably keep this thing together. Like you're not going to expect that your guide RNA sequence is actually distributed across the whole plasmid. Like reasonably keep it together. And then I assign these gap scores. So it does allow some gaps, but you can give how much the penalty is for those gaps. So this is the open gap score, which is you basically lose 10 points of alignment if you start a gap, and then every base pair you extend it, you lose half a point of alignment. So this allows for, some sort of gaps. So for example, when I first did this, if you don't want to make a map for every single guide RNA, you could use one and align all of your different sequencing results to that. So it will still align, but it will show a misalignment at the guide RNA cloning site of those 20 base pairs. So it'll lose some points for that, but everything else should align. So then it will still work well. Um, I then, since we still haven't like accounted for this reverse complement thing, I just set a variable called reverse complement bool for Boolean and set it to false. And then I want to get what would be the alignment score to the forward sequence and to the reverse complement sequence. So I'm leaving the reference sequence the same, but either I'm taking the new sequence or the reverse complement of it. Again, we can just do dot reverse complement and see which score is higher. So if forward score is higher, we want to just use the forward one, and I'm going to set RC bool to false, um, or really keep it false. And if the reverse complement score is higher, I want to set it to true because we want to be focusing on the reverse complement of the new sequence that we got. So then to do the actual alignment, I'll do aligner.align and the reference sequence and the new sequence. And then this will actually do multiple alignments, but the highest scoring alignment will be the first one, which in Python is the 0th one. So I'll just use these brackets and the 0, and then either do it with the forward sequence or the reverse complement if that ended up having a higher alignment score. You can then print this alignment, and it looks a lot like what you might have seen in other software. I didn't use the whole thing because Gearbox is like 6,000 base pairs. But you can see here that like my whole plasmid sequence matches this reference. The coordinates of the reference are quite a bit different. So the 0th item in my reference map is the 3,139th item in my new sequence. Plasmids are circular, so it really doesn't matter what you call the beginning. But you can see there's a line for everything that matches, and then there would not be a line if there was a mismatch. This had no mismatches because it matched my sequence. But yeah, that's a way that you can do this alignment in Python, and you can imagine since you're getting a quantitative score, it can let you really quickly screen through a lot of sequences in a quick way. Any questions on all that stuff so far? So now that we verified that our plasmid is actually cloned correctly, you would then test the guide RNA in a cell type of interest. This can be done a lot of ways. You could do it in a cell line as long as it's from the same species that you're planning to target. You could do it in your actual species, like in an animal, but target all of the cells, which is a little bit easier. Or you could target, for us, when you're doing like an astrocyte specific in vivo editing, you could just start with that and verify it that way. So really up to you. We're going to assume that you're experimentally perfect. So we're just gonna go ahead and cross that off. You did the experiment great and it worked well. So now that you have material, the easiest thing that you can do with CRISPR is to say, did I actually have mutations in the DNA? Since that's the actual mechanism of CRISPR-Cas9. Downstream things could also be really helpful, like reading out, was there a decrease in the RNA for the target, or was there a decrease in the protein, which would probably be the functional outcome. However, those things are usually a lot more difficult, and they're usually, target specific. If you want to screen a lot of different CRISPR tools at the same time, you can always go back to the DNA. So that's usually the first pass that I would do. So the way that we do this is through a PCR strategy to amplify the guide RNA target region. You could obviously sequence the entire genome of the animal or cells that you care about, but that's going to be extremely expensive. So what we're doing here is just PCR amplifying only the region the guide RNA is supposed to be targeting, and then sequence that. So I have, again, Colab code for designing those sequencing primers. So the way that I do this is by first downloading the actual genome assembly for the species you care about. So for this, I chose the mouse MM39 genome. These are available from a lot of different sources. Here I got them from Ensemble, but they're also on like UCSC and other things. I then use command line g unzip command, which basically just removes this .gz and like unzips that. There's a lot of other things that I need to install here. So we have Biopython. Primer 3 is one of the main algorithms for designing primers. pretty much anything you use is probably using Primer 3. So Primer 3 has websites online. NCBI Blast Primer Design tool also uses Primer 3 under the hood, but here we'll use it directly. And then Beautiful Soup is something I added here that allows you to use some web tools that I've mostly removed from this because they stopped working. Yeah, the previous iteration of this used UCSC's web tools I talked about earlier. They have since made it so that they block algorithmic access to them. So those don't work anymore in this way. They still work great if you're a human on the website, but they don't work for Python. Um, so first, reading that genome file, or just saying the path, and then I'm going to turn that into a dictionary. The basic way that this is working is that the FASTA file for a genome assembly is obviously, it's going to have sequences for each chromosome, and there's going to be a break, right? Because the chromosomes are not, continuous with each other. So by making it a dictionary, you're making it so that you can quickly grab an individual chromosome instead of having to like look through the whole file every time. Um, I did I zoom in on this? No. I then go through each of the guides in that guide list, and I'm going to make that my query. So I'm going to get the forward and the reverse again because your guide RNA, might match to the reverse strand of the genome assembly. So we want to check both of those. I'm then just going to search for each chromosome in that genome dictionary. Um, and then the sequence will be this record.seq, which is just the sequence for that individual chromosome. I was trying to scroll, but I can't. I then am going to use for each query sequence in this list of the forwards and reverses. I'm going to like search for those iteratively. So first I'm trying to find what is the start position where this guide RNA matches to the genome. So that we can use the sequence.find thing that we were using earlier. So we're just saying in this sequence, which is a chromosome, um, where does this query arrive? So, or match. So once you actually find one, we'll then say the in position is the start position plus the length of that sequence. Basically always 20 base pairs for CRISPR with a couple exceptions. We then want to get the surrounding region. So for this sequencing, we use GeneWiz's Amplicon Easy service. So that requires a sequence that is less than 500 base pairs. But we want to give ourselves a little bit extra room to design the primer, like one way or the other. So for this, I'm getting basically a 500 base pairs upstream and downstream of the guide RNA targeting site. So to start that surround, we want to get the start position minus 500 and the in position plus 500. And I have this max with 0 and min with the length of the sequence, just for the edge case that your guide RNA is at the end of the chromosome in the genome assembly, because that will like make things very difficult. So if it's at the end, let's just use the first nucleotide, and if it's at the other end, we'll use the last nucleotide. You'll have slightly less region around. So if you're literally targeting like the 0th position in the chromosome, this probably will have to adjust things. But in any case, you then have that surrounding sequence. And then we want to append that to the sort of output, which is this match data thing that I created. So we'll have the chromosome, the sequence, and then the strand of the actual chromosome that it is. So if it was from the forward direction, it's the positive strand. And if it's matching to the reverse complement, it's the negative strand. Yeah. So this is giving us that genomic context that we can use to design the primers. Next, we'll do the actual primer design. So here we'll just use what were all of those matches that we found. I guess I didn't write it in here. We'll get to later. If there's no match, we want to exclude those. Also, if there's no match, your guide RNA is really bad and will not work. So that's probably one thing to do at the beginning before. All of these tools should be doing that. But I have seen like these If you go to papers, some of them have like a list of guide RNAs that are supposedly for like every, mouse gene or something like that. I have found some of those don't match the mouse genome at all. So be aware that might happen. Um, so for primer three, basically we start with this huge design primers like function, and it takes a bunch of arguments. If you go to the primer 3 website, these are sort of the arguments that they have there, which I was trying to emulate. For some reason, the defaults on the website and the Python version are not the same. But since I've used the website a lot, I just wanted it to be the same as those. But they're pretty sort of standard things. We want the primer to be 20 base pairs. It can be, you know, minimum size 18, max 23. We're going to look a lot at the melting temperature for the primer to show like how it would anneal to DNA. We can give a range of what temperatures are ideal and then what are acceptable. GC content is, again, another important thing for primer design. So we're going to give a range on how those can be and kind of et cetera. We also want to say what is the product size range. So for this, I made it 200 base pairs to 467 base pairs. Um, that is because the maximum product size that GeneWiz will take for the sequencing is 500 base pairs. I believe they're doing 250 base pair parity in sequencing. So if it was over 500, you wouldn't see the middle, which is why they require it to be that way. And 467 because you require a sequencing adapter that we'll add on later. So that's going to add a little bit of sequence. And then for the actual target, we're going to take that string that we produced from searching the genome earlier. So that's that whole 1000 base pair region. And then for target here, I want it to be within that sort of middle chunk of the sequence. So basically, this is saying actually include that middle piece because that's where the guide RNA target is. So it's not going to just make, if there's really good primers for the first 200 base pairs, you would miss the guide. So this target thing is constraining it to be there. So it's going to have to look starting at 440, which is around the middle, and then 140 it needs to include the 140 base pairs that are after 440. So it gives you like a nice region around the middle, which should be 500, like the 500th position, basically. I guess 500 to 520 will be the guide sequence. All right, so then once you found this, I just like to double check that you'd actually produce a good product. So for this, I just take the left sequence and the right sequence out from the primer 3 output. It has this sort of syntax where it says primer left, the index, and then the sequence. When you run primer 3, I think it makes 5 by default. So this is basically just iterating through the first 3 and checking them. And we're just basically doing that same find thing that we were doing before, where I'm checking in the actual genome. I guess I defined this function. Yeah, I defined it here. That'll make it easier to see. Basically, I'm just searching in the genome, is the forward primer and the reverse primer matching to the genome? And what's the length between them? Is it actually making a product of the acceptable size and all of that stuff? So this is just sort of a double check that is probably not necessary, but I like to have for assurance. I think I have a duplicate slide. So that's running in that function that is on the next slide. And then basically just return the first one that is matching our requirements. All right. So then I take them and I add, these are the partial sequencing adapters from GeneWiz. So this will actually allow this DNA product to bind to the sequencing chip so that it can do that paired end next generation sequencing without any sort of like library preparation. You're basically just doing the PCR and you're done. Like they can directly put that into the sequencer. The other one I have here that I haven't used too much is sort of an alternative if you wanted an earlier check that's a little cheaper. Here I added the M13 universal primer that GeneWiz has. So if you did this, you could make this PCR product and then send it for Sanger sequencing using that universal primer. The next generation sequencing is in the neighborhood of $60 per sample. The Sanger sequencing is usually like $5 per sample. So if you wanted to do an earlier check that things are working, you could do Sanger sequencing. And the quality of the Sanger sequencing alignment or like peak calling, um, again, I guess it's probably not the right word, but base calling will decrease at the cut site if you're doing Sanger sequencing. Um, in our experience, that doesn't like it's kind of hard to see. Like you wouldn't be able to very easily quantify the actual indel percentage. Whereas with next generation sequencing, you get an actual percentage and you know actually like what is the sequence of those mutations? Are they going to be frame shifts and that sort of thing? Um, in the end, I then save this to a file and export that so that you can order those primers from IDT, which I think I excluded from here, but same as before. So now that you have this PCR strategy, you would then PCR amplify the genomic DNA that was edited, and then you should be able to validate this, right? I guess there's a meeting after, but um, you would then do that PCR and send it for sequencing, and of course, um, we can then validate that that's correct. So you can't do like a simple alignment. If the alignment works really well, you probably didn't get any gene editing. And you also would have, a lot of sequence, like thousands of sequencing reads, so that wouldn't really work. So the tool that I really like for doing this validation is called CRISPR. They have a web version and a command line version, which is a requirement for any tool for me. Um, so I also really like that they have these cute like coffee animations everywhere. When you run it, it actually like the loading bar is a cup of coffee like filling up, which makes me really happy. So it looks like this. So you can give them, say that you use Cas9, if you do this paired end sequencing that I talked about, and then you would give them the forward and reverse sequencing files, which GeneWiz will just get back to you. You then would say the amplicon is like, what is the sequence of DNA between your PCR primers in the genomic like wild type case? So if there was no edit, what would the sequence be? That's what it's going to align to. And then you give the actual guide RNA sequence. Sometimes you'll see S guide RNA. Those terms are interchangeable. And then for the command line version, I didn't go through how to like install that. It doesn't work well on Colab, or I at least couldn't figure out how to get it to work on Colab, so I use it locally on Linux. But once you have it installed, which you can see they have instructions on their GitHub page, you can use this Crispresso batch command. And then basically there's, oh, I just didn't highlight them. It's basically the same inputs, right? So you have batch settings, you make this file called batch. So what this is going to do is actually run on all of your files, whereas the website only runs on like one sequencing result at a time. So if you did a lot of sequencing, from like multiple animals with the same guide RNA, this would be easier to use. You then give the actual sequence again of that target region. The capital letters, which it's not reading at all, but is how I like to have these, is the actual primer sequence. So you can see that you include the sequencing or the primers that you did for the PCR without the adapters, and then you have the actual sequence there. -G is the guide RNA, and then it will return either if you use the website or the command line version, something like this. So you'll get a percentage of sequencing reads that were modified. You can see here 38,943 reads were modified and 21,000 whatever were unmodified. The other thing I really like about Crispresso is you can get a plot like this where this dotted line is the predicted location of the double strand break, which is a few base pairs to the left of, I think it's 3 base pairs left of the PAM sequence within the guide RNA sequence. And then you get a plot of the number of deletions and insertions and substitutions at that locus. So you can see here that like the gene editing is very specific to the region that we targeted. This also confirms that your PCR was good because if you did PCR, poorly or with a low fidelity polymerase or something like that, you would see a lot of substitutions like just everywhere. But we're having very specific gene editing at the target location. So that's something I really like to see to know that things are, very specific. So that's sort of the end. These are kind of those overall steps. So whether you do it with Python code or just, conceptually with these different websites, I talked about designing the guide RNAs to the target of interest, ordering those oligos for cloning them into a backbone. You can verify your cloning with plasmid sequencing of some sort. You then would test the guides in a cell type of interest, then can do PCR to amplify that target region and validate that through sequencing. After that, of course, then you probably need to do like the experiment, maybe validate through protein or RNA. RNA is often not heavily affected by CRISPR, so probably through protein. But yeah, this is at least a pipeline you can use for every target before you have to get to the nitty gritty of seeing like an antibody for your target and things like that. Yeah, but that is it. Any questions?
Did you code upload it on GitHub or?
It's not on GitHub, but I have all the links in the slides and I have them like
To all of the like,
shareable.
show?
Yes. I guess I don't have anything for the Crispresso, but I mean that's there in the slides, pretty straightforward. And then I didn't put anything for guide scan because I couldn't figure out how to get that to work on Colab yesterday. But I do have the Gershbach Labs papers there, and they have like a step-by-step. So the other thing that I sidestepped, they have a new version of GuideScan, and that's why I couldn't figure it out. I've only used the previous one, and I couldn't get the new one to work on Colab. But they have detailed instructions for using the previous version, which is also still like available on GitHub. The new version like makes it harder to make guide RNAs for some reason. I don't know. Their command line interface just like acts like that's not what you're using it for. It's mainly like written as if you were using it to check for off targets, which is like part of the pipeline, but it doesn't, it seems less straightforward for some reason. But yeah, those two things I don't have code for, but all of the Colab links should work. And I have them like so that anyone with who clicks the link can view it, but not edit it. So then you can copy it to your own Colab and all that.
These slides, how do we get access to?
Yeah, so they will all be, I guess I'll just take there. We have all the slides and we'll have the recording on our qlscenter.duke.edu website. So if you go qlscenter.duke.edu, resources here, we have all the talks here. So it'll show up as the next bullet point. And we'll have the slides are the link that has like the name, and then the video will be attached here of this recording.
And so like you had the reference in this slide to like a previous.
Yeah, so
resource that
then,
will
yeah,
also-
yeah. So the links then you can get from the slides and click and all that, and they should all work.
Like,
Quick clarification.
Mhm.
The Crispresso tool,
Yeah,
the amplicon sequence that you're using is the same on the PCR Sanger sequencing amplicon? Or
no. So the Sanger that I showed earlier was from the plasmid, and this is like in the genomic DNA of the like edited region. Yeah. Yeah. So you could do a Sanger sequencing of the genomic DNA as well. That's what that like M13 forward would be. And then you would just see like the decrease in Sanger base calling at that location potentially. But you would need to use a different primer set to have those sequencing adapters for next generation sequencing. And in my experience, it's really hard to tell from Sanger the indel percentage. So I would just go with the next gen, unless you have like a ton of guides and not enough money to do the next gen. Oh,
thank you.
we'll stop sharing. Hopefully there were no questions.